Tracy, and I'm a professor of mechanical engineering at the University of South Florida. And hopefully you'll understand by the end of the talk what I'm doing at the conference like this. So the title of my talk are Human Ropeworms Description and Current DNA Findings. In January of 2013, we tried to publish a paper in the scientific literature, and I submitted the first paper, which is called Human Anaerobic Intestinal Rope Parasites. I submitted this to the International Journal for Parasitology, and they rejected it without review. And the editor said that there has to be a scientific proof that this is a parasite, and when I asked him what's the proof, he said you got to do electron microscopy and you're going to do molecular studies to prove that this is a parasite. And there is a very important emotional aspect of this. When chronic symptoms that a person might have had disappear like this, and it's a very affecting emotional experience. So as a result of that, I approached Dr. Gubarev, and he told me what's not written in the book, including other methods, and they're really, really involved. So I said, we've got to write a paper about it, because that's a part of my job, is to write scientific papers, and I did. So these are the papers that were submitted to arXiv, which is a database made by Cornell University, and there's no peer review for that. That's how prominent mathematicians deal with their papers. They just submit it out there and say, we're not going to deal with the peer review process, which I'm a fan, because you know, I'm a professor of engineering at the University of South Florida. So the second paper was written, that's called The Developmental Stages of the Rope Human Intestinal Parasite. And this is actually the reason why you listen to this talk, is because Joe contacted me, the organizer of the conference, and invited me to give a talk here, including Dr. Gubarev. So if not for these two papers, there would be no stream of data and there would be no this talk. So as you can imagine, I'm not a medical doctor. My wife is an MD. She has a degree, an MD and a PhD, specialized in endocrinology, from Russia. So I would like to put some relevant Wikipedia definitions that most of you are probably familiar with, but they're going to be helpful for this talk. So homeostasis is the process in which the body internal environment is kept stable. And when the body is out of homeostasis, then symptoms may develop. What's a parasite? A parasite is actually an organism that has sustained contact with another organism to the detriment of the host. But this implies that these are separate organisms. So like if you have a macrophage, or if you have a mitochondria in the cell, is that a parasite? Parasitism is a non-mutual relationship between organisms of different species. And I underline the important things, different species, where one organism, the parasite, benefits from this, benefits from the host. Now there's another thing that's quite common in biology. It's called symbiosis. Symbiosis is an interaction between, again, two organisms living together, an intimate association or merging of two dissimilar organisms. So does husband and wife qualify? A worm. A worm is an elongated, solid-bodied intravertebrate animal, most commonly the earthworm. That's the definition of a worm. And there are also parasitic worms, and the term for that are helminths, and they are eukaryotic parasites, meaning that they have many cells, versus prokaryotic like bacteria. And if they are in the intestinal tract, they're called intestinal parasites, and we know that both humans and animals can be affected by them. A polyp. A polyp is an abdominal growth of tissue projecting from a mucous membrane, and it can be either attached or not attached. But there's this connection with the mucosa and mucous membrane. So the question is probably to a biologist or somebody who understands how nature works. Can a parasite be produced? by the host itself or from the host cells? Is this really possible? Okay, so you've seen these pictures. We gave it a Latin name, which is a Homo Funis Vermis, human rope worm. This is how they look like, and the pictures that you see are from Dr. Gubarev's book. They are meter and a half long, slimy looking, and they are anaerobic, meaning that they dry up really fast. If you get rid of them and they're sitting over here, they dry up. And if you want to store them, the best way to store them is an alcohol, is an ethanol 70%, and if you don't have that, vodka would do as well. So Dr. Gubarev patented 3 different methods to get rid of them, and he's written a book in 2009, and the patents were awarded in 2007. He actually started his research in 1998. So what is this? What is this stuff? Is it mucosa-associated lymphoid tissue? Well, what is mucosa-associated lymphoid tissue? It's a diffusion system of small concentrations of lymphoid tissue found in various sites of the body, like the gastrointestinal tract, thyroid, breast, lung, salivary glands, eye, and skin. Okay, that's the definition from Wikipedia. That's what MOLT is, mucosa-associated lymphoid tissue. And the doctors specified if it's found in some other, they can say it's nasal, nose-associated lymphoid tissue or vaginal-associated lymphoid. So whenever they find it, is it a parasite? Is it both? Or is it, as suggested, as a lining of the intestines? So we will try to answer this question. These are the microscopic observations, optical microscopy. And what you see here on the left upper slide is the image which shows a scale-like appearance. These scales are actually cells. So what you see here are the individual cells, and here's the nucleus, and in the next slide I'm going to show you that this is really so. Inside the microchannels in the body, there are bubbles, they're gas bubbles, and here's one blown up. So they have cellular structure, and they're gas bubbles in the microchannels. This is a DAPI stain. DAPI stain brings out the DNA in the nucleus of the cell. So these are the cells, and that's the DNA. All right? So electron microscopy, you got to do this in a special microscope, and by training I'm a microscopy, so I know quite a bit about this. We try to look at the surface of the rope worm, and because of the vacuum, it's low vacuum, this whole structure crumbles and buckles, so that's why you see this appearance, but these microchannels terminate on the surface. Okay, here's a hole that goes inside. And with an SEM, with a scanning electron microscope, you're looking at the surface. There's no really a way to look inside. Unlike maybe with some optical microscopy. The previous DAPI stain, that's a confocal microscope done with the staining of the sample prior to that. So the earlier stages that Dr. Gubarev described, and based on the morphology classified them, they look like mucus with bubbles, gas bubbles, and sometimes there's blood. I've actually talked to a specialist, several microbiologists and biologists, and they say that it's quite common that parasites attach to the lining of the intestines. It doesn't necessarily mean that they feed on blood, but I think Dr. Gubarev, with his pathology friends in Russia, has some evidence that maybe this is the case, that they do feed on blood. These are the earliest stages. Here's this transition that he showed of the jellyfish to the seaweed kind of thing. And here's another one, which is the branchy, bush-like. So you've seen these pictures, and I'm going to skip for that. The fecal stones. These things either originate from the fecal stones, or they produce fecal stones. And Dr. Gubarev thinks that they originate from the fecal stones. I was under the impression that they produce fecal stones. So here's a sample. You can see it is kind of full in here, and it's not full. Here's the body of the worm, and here's a fecal stone.
So what is this fecal stone?
A piece of fecal matter that has white dots on it that look like seeds. And this is a repeatable phenomenon. It's not just from one person. If you're ever going to see a fecal stone, that's what it's going to look like. So you can see this fecal stones, and you can see the mucus stream, or whatever you want to call it. Okay, so at the DNA was extracted successfully, and the sample was collected during water-only fasting to avoid any contamination with food, because that's the problem. Okay, so here's the stage 3 and the stage 5. This is the DNA strand that was extracted. This is a gel, and some parameters of the extraction that you see over here. And they said, from one of the journals, they said you got to do DNA barcoding. What is a DNA barcoding? Well, they've taken the cytochrome oxidase gene, and they've taken the side of that has about 600 base pairs of this mitochrome gene. But what is this thing responsible for? Well, it's the breathing. It's how the things breathe. They convert molecular oxygen to water. That's what CO1 gene is responsible for. So there's probably a way, and there's some controversy in the literature whether or not it can be used for barcoding, but people have tried to do that successfully. So this was an initial attempt, and I'm not a biologist, so okay, so here, what's the test? Here's the test. Okay, let's do the test. For this test, we used polymer primers that are not 100% specific to any particular species. These are the primers right here that were used for the sequencing done from the extracted DNA, and these are the sequences. So if you go to NIH site and you blast them, the stage 3 comes down as a 90% human pseudogenes chromosomes 8 or 17. They're equal, they're the same sequences. And interestingly enough, like number 5 on the blast is chosen as a 98% Simoninus notabilis, which is a seed bug. Okay? And the stage 5 is a human mitochondrial DNA. That's what was measured from the sample. Now, if you exclude the human and chimpanzee genome from the blast, you're going to get a mite and you're going to get a worm. And this sequences only match 82.6%. Okay, so whatever that means, their CO1 gene is not the same for the stage 3 and the stage 5. Okay, so why would I pay attention to this? This is the insect. During the conversations with Professor Gubarev, he told me that a very strange thing happened. He used to photograph everything, and he left those fecal stones for about 5 to 6 hours. And this is St. Petersburg, Russia, in spring. He came back, and they were covered with insects that he has never seen in his life, ever. So once I saw this result, I went to Google and I collected images of insects of various kinds, including this one. And I sent it to him. I said, Dr. Gubarev, which one did you see? He said, I would never forget it. The image is in my head. This is the one. Okay, so he saw this thing. This is a tropical bug that eats rice in Japan. Okay, this is a tropical bug. I have it in my house in Florida. I was never able to see them directly produced out of fecal stones, but that's what happened, and that's why I'm showing you this. Okay, this is a mite, and this is a worm that lives on. So again, this is 100% match to the partial sequence of that gene that was sequenced from that. Okay, now biologists and people that did this, they say, Alex, this is human DNA. So what's the next step? The tree of life, the gene 18S, and that's what the scientists use to look at the evolution and the history of organisms. They can also reconstruct ancient divergences from this, and that's how they do phylogenetic studies, based on the 18S gene. And again, what's important is that you use universal primers, meaning that those primers are not specific to any species. Okay, there's a universal primer. These are the sequences. 99% human 18S RNA. interestingly enough, 99% uncultured fungus or uncultured eukaryote clone. 18S sequences match 99.3%. So again, results are inconclusive, and there's going to be a Illumina run. Actually, it finished on Friday. The data is being analyzed. I have not seen the data. Match to human DNA is 28% so far. So, where did these guys publish this? Well, you can see the sequences. All right, this is the human 18S RNA, and these are the 4 sequences. They did the forward and reverse reads. That's why there are 4 of them. Okay, stage 3 and stage 5. This is the NIH data from NIH GenBank. So this is published in Nature in the spring by the NIH Institute of Human Genomics or something like that. So Nature. I contacted the author, the corresponding author, and I said, the paper is called Topographic Diversity of Fungal and Bacterial Communities in Human Skin. I said, the sequences submitted to GenBank also match human 18S RNA gene. How do you know that the sequences belong to the fungus and they're not human? And the response is, you're right, they are human. What about Cytominus notabilis? Again, experts in genetics, they tell me that sometimes you don't get the PCR amplification, so eventually the sample gets contaminated with human DNA, and he thinks that he sampled the bug and it was human DNA. But the question is, how did Dr. Gubarev see this seed bug in St. Petersburg? That still remains a mystery to me. So, okay, YouTube, great thing, allows an engineer to look inside a human body. So the first thing is a colonoscopy. And there are tons of pictures and videos submitted by medical doctors from different countries that administer colonoscopies. And the narration for this one says, oh, this stuff, oh, that's normal colonoscopy. This is lymphoid aggregates. This stuff and this stuff. It's actually from the same video. The inventor of colonoscopy, Dr. Hiromi Shinya from Japan, has an instructional video of what colonoscopy is. You see this thing here, you see the vessels here, and this stuff here is the ropeworm attached to the intestinal wall. On the video you can clearly see this. This is a polyp removal from Russia, and you can see that right there at the polyp site before they burn it off. There's the same thing. And you can look at that yourself. Just Google colonoscopy. All right? Bronchial tubes. There's mucus in there. Well, that's what it looks like. It resembles the mucus that maybe looks like the early stages of the ropeworm. Here's the video. You can watch the video. And nose is the most interesting one. A lot of doctors from India like to post their endoscopic operations, all sorts of polypectomies and sinuses relief and things like that, and you can see this in the sinuses. Here's this bubbles and mucousy thing, and they cut tissue out, but behind that, that's what it is. There's one precious one. If we have time, I'll show you the video. Another guy in India was leaking through his nose, so they put a dye into him, and they saw it leaking from the nose. They wanted to figure out where it's leaking so they could stitch him up. Okay, so here's the dye that appears with the bubble. And doctors call it nose-associated lymphoid tissue. No. All right, you can have a discharge from vagina. That will be called Walt, and so on and so forth. Now this is an interesting. This is my favorite. Okay, it's a 2 feet long thing being pulled out of a nose by an endoscopic surgeon. And if we have the time, I'll show you the video. So here's the nose of a patient, and here's the thing. He calls it inspissated mucus. Now, the send it for culture comes as a Staph aureus. Okay, is it Staph aureus? Do you believe this is Staph aureus? What is it? Inspissated mucus. Great. So Dr. Gubarev calls it a seaweed rope worm, and you call it inspissated mucus. You don't know what it is. I don't know what it is. Seriously, this is the situation, okay? So, nasal polyps. I gave you the definition of a polyp. What's a polyp? It's just a big snot, okay? That's what it is, a polyp, okay. Google image. This is the polyp in the nose. Here's another one. And this is a coughed up substance after swimming in the Gulf of Mexico. This stuff comes out, a big snot. Okay, nasal polyp, great. Appendiceal Mucocele, it's such a rare thing. Doctors publish papers about it. You cut out the appendix, see what's inside, and there is Appendiceal Mucocele. What is that? Well, here's a video. Here's a link for that. Morphologically, it is mucus with bubbles inside. But those of you that have done surgery, this is what most likely happens in the appendix. The appendix gets inflamed because of the fecal stones inside the appendix. Here's another video of that. Okay, so this thing gets into the appendix somehow. The appendix gets inflamed, and then the surgeon removes it. That's not the end of the story, though. I had no clue. Okay, I'll tell you the story in the end, what happened to me, why I'm who I am and I look like I do. I thought it's all in Russia. So in Russia, there's this forum that has over 1,000 posts to it. And basically, the patents that Dr. Gubarev has in Russia, they're out there in the open literature. And the community of these healers or whatever those people are, they decided, oh, great, this is called ELM, Eucalyptus Lemon Method. Let's do that, and let's make money doing that, selling people brooms of eucalyptus leaves and eucalyptus oil, and there's a website, they can sell you the stuff, and people send them feedback. Oh, look at this. Look what I got. I caught some big fish. Over 1,000 posts. And there's some more Russian forums, okay? Because of the papers that I submitted to archive, I know that this person is, this is universal. On curezone.com, there's a person every day submitting a picture of this, except that in the United States you wear a glove, okay? Right? So these are the 3 websites, the forums where people share this, and I was completely unaware of it. And more. And more. And then there's this group that deals with autistic children. And when the first paper was written, we said it's going to take 40, 50 years for this thing to develop. No. These children are very small. But they do administer enemas, and that's how they clean their bodies. And they have, whatever, 80-something kids completely recovered from autism by this enema cleansing procedures. And they have a lot of pictures. And they gave me access to those pictures. Okay? So that's what you see. You see a rope worm, Homo Funis Vermis, or mucosa-associated lymphoid tissue, and a fecal stone that you can also find in appendix and elsewhere. And the Andreas Kalker is the leader of that group, or he's associated with them. This is a picture of a similar thing that was sent to me by somebody who read the paper. And this is the herbal cleansing that she did. She didn't do an enema, I believe. I didn't even look at that, what that thing is. She said, my child is sick, 5-year-old girl, she's autistic. I gave her that, and this came out. And it looks exactly like what you describe in your paper. Look at this. The medical community cannot ignore the fact that this thing exists. Okay? This is it. This is the end of it. Inspissated mucus, tell me what it is. Is it normal? Is it not? What is it? Is it the part of our buildup? Is it mitochondria that produces the energy for us, and that's the payback that we have to pay for all the food that junk food that we eat? I don't know. This is out of the video. Okay, another one. A guy did a juice fast. That's what came out of him. Another person who contacted me. Juice fast. Oh! Another picture from the autism group. A stoma. And it's not just one, okay? So the intestines were removed surgically, and it's an unfortunate situation to be in, but here you go. You see that? And a database. I mean, they have it on Dropbox, and I'm grateful that they gave me access to that. 6 patients, or 6 cadavers, examined in Volgograd. There are 2 cases here, 74-year-old male who died of heart attack, and another guy who died of pneumonia, late age. What actually happens, if you have a pipe, if you have a tube, whenever you use any sort of an enema procedure, it comes out intact. Once you cut it out, you relieve the pressure and this thing bursts. So it looks different. If you cut the intestines out and you make a section. And you can see this is a more or less even surface. If you look at the videos of colonoscopies, diverticuli, what's the name? I mean, it goes like this. Yeah. All right, you see that? Once you cut it open, it doesn't look like this anymore. It looks like this. So, I'm a scientist and an engineer. Here's the data. 54 people as of yesterday. I received 2 emails yesterday. These people, since January of 2013, fall into 4 different categories. People that say, oh, I'm so sick, this is it. The rope worms are killing me. They're eating me alive. Parents of autistic children is another group. So 34 of those. 8 parents of autistic children. Lyme. And 2 people with Morgellons disease. What else do they have? Well, they have Crohn's disease, they have diverticulosis, irritable bowel syndrome, daily rectal prolapse, whipworm, other things, candida, fibromyalgia. So, is it a parasite or is it human? Well, one's gone, they don't come back. If it's human, if we depend on them, then why don't they come back? They cause symptoms, whatever those symptoms are. And the symptoms are relieved almost instantaneously. You have to experience it yourself, which I have. They move inside the body. They have microchannels with gas. They leave the body with enemas. There's no blood or tissue. In the tissue, there's no blood. So blood does not go in there, so suggesting that maybe this is a foreign object in the body. They absorb food. There's fecal stone connection. I don't know if they make them or they come out from fecal stone. They produce toxic mucus. They do increase blood pressure. The pressure goes up by 20 points while you're holding an enema. It's like, hard to hold it. They stay in the intestines even when they're dead because they have this mechanism to attach. They're sticky. Well, but maybe they're human. Could they be human? Well, so far we sampled human DNA. And the experts say, oh, this is human DNA. Mold, mucosa-associated lymphoid tissue. Well, do we really know what it is? There's not much literature about it, seriously. They've done some studies on mold in some police people in Italy to see what's in there. One thing that if a muscle test, many of you know what a muscle test is. Chiropractors use it all the time, the biofeedback test. A ring test, an MD from Japan, wrote a scientific paper about it. Why do I bring this in here? Because the one person does not necessarily get affected by those rope worms from the other person. Sometimes they do, sometimes they don't. So actually, this last one can go both ways. Okay? Now, in conclusions, this is what I can say. This ropeworms do leave human body when you use various cleansing procedures. And you can use pharmaceutical drugs. You may see them. You may use alternative medicine. You can use different enemas. You can take other things that were described by Dr. Gubarev. He has 3 patents for those methods and a book. There are hundreds of people that pass them out, if not thousands. These people feel instantaneous relief of the symptom. The symptom gone, like this. In a moment, they leave the body. This ropeworms either produce or originate from fecal stones with white dots on them. It's a repeatable phenomenon. The 2 genes sampled, the CO1 and 18S, is probably human DNA. Are we mixing the cause and effect? There's some theories that we've heard in previous talks that maybe bacteria is a marker, not the cause of disease. So maybe this worm is just an advanced trash bag. I don't know. It could be. I mean, I can only speculate at this point. So what is this really? The definition would be this is mucoid, worm-like aggregates, and the origin of them is unidentified at this point. So what's the way to solve this problem? Sequence the whole thing. So what's the problem? Funding. So the Chronic Research Disease Foundation is trying to collect funding to do this. And you're talking about a substantial amount of money to sequence the whole gene. Luckily enough, the United States of America is leading in this area, and this is where you're going to do it, and that's where it's going to be most cost efficient and cost less compared to other countries because the technology comes from the States. Okay. So